This week we're going to be looking at DNA, and this is going to basically expand on what we've already learned about DNA in terms of it being one of the 4 types of organic molecules. So we're going to look at things of more specific information about the structure of DNA, and then we're going to look at what does DNA do for us and why is it contained within the nucleus of cells. So the first thing that DNA, we're going to talk about with relationship to DNA is what its basic function is in the body. And at its most basic, you can think of DNA as simply being a recipe or instructions for the making of proteins. And if you look at a long string of DNA in the body, called a chromosome, and we as humans have 46 chromosomes, but if we look at just one of those chromosomes, that chromosome is a long string of DNA. And on that long string of DNA, there are different genes. So here I'm drawing in blue here. Here might be one gene, here might be another gene, here's another gene along the length of the chromosome. And each of those genes is one recipe or one instruction for the making of protein or a collection of closely related proteins. Okay? So each gene is a recipe or an instruction guide for the making of a protein. Now we'll go into explaining exactly how those instructions code for the making of a protein in a couple weeks. But for now we're just going to learn that a gene is a recipe for a protein, and that the purpose of DNA is to serve as sort of the cookbook or the storage, information storage facility for all of these different recipes for making proteins. Now the way in which DNA and its function in the cell was discovered is kind of interesting. Your book talks a little bit about it. This is from an experiment by a guy by the name of Griffith. And what he did was he had some mice and he injected them with a certain bacterium that in its S strain, that's just one version of the bacterium, was very deadly to mice. So when he injected the mice with the bacteria from the S strain, the mice died. Now, when he would take a different variant of the same bacterium called the R strain of the bacterium, and he would inject it into mice, the mice would not die. And Griffith believed that there was something about the fact that the S strain bacteria had this gel-like capsule surrounding the bacteria, and the R strain bacteria did not have this gel-like capsule surrounding them. It was something about that capsule, he thought, that made the bacterium deadly, so that the mice's immune system could not combat the bacterium. And he postulated that the DNA inside of each bacterium coded for the production of a capsule in the S strain, and that recipe for the making of a capsule was lacking from the bacteria of the R strain. So what he did was he heated up the deadly S strain bacteria, and what that did was it basically denatured the proteins, and it also denatured the DNA inside the S strain. And then he injected those denatured bacteria into the mice, and they lived. So that suggested to him that, hey, DNA might have something to do here with the production of the capsule, with the recipe for making the capsule. But just to be sure, he then took some of the R strain, and remember the R strain bacterium did not make a capsule, and did not have the DNA for making a capsule, and he injected both the R strain bacteria and some of the heat-treated deadly S strain. And what he found was when he injected those both together, the mouse died. And what he postulated, and what was later shown to be true, is that the DNA, the inactive DNA from the gel-producing S strain, was taken up by the R strain in a process called transformation. And so that DNA was transferred from the dead S strain bacteria to the live R strain bacteria, now that the R strain had the recipe for the making of a gel capsule around it, they too became deadly just like the S strain had been. So that was basically kind of the background for how it was shown that DNA indeed does code for the making of proteins. Now we can go on further and talk a little bit more about how another experiment also helped us to support this idea that DNA is actually the instructions for the manufacture of a protein. And that involves a very brief discussion of the Hershey and Chase experiment. Now in this case, what Hershey and Chase were trying to show is there had been a debate going on in science over whether it was the proteins inside of an organism, of a cell, that basically directed activities of that cell, or whether it was the DNA that was the director of activities. And so what they did was they looked at viruses. And if you don't know, viruses infect host cells by, we now know, by injecting DNA into the host cell, and that DNA that's injected into the host cell then begins to instruct the cell to make more viruses rather than doing the normal cellular activities. Now at the time, Hershey and Chase didn't know that. In fact, scientists didn't know if it was the protein part of a virus that was the infectious part that caused damage to a host cell, or if it was the DNA part of a virus that was that caused damage to the host cell. So what they did is they took some bacteria, and they took a special type of virus that infects bacteria called a bacteriophage. Okay? Any virus that infects bacteria specifically is a bacteriophage. So they took these bacteriophages that had their DNA radioactively labeled. As you can see in this picture here, they've made the DNA yellow to indicate it's been labeled with radioactivity. And they allowed the viruses to infect bacterial cells, and then they put the virus and the bacteria into a blender and mixed them around vigorously. And what that did was basically dislodge the viruses from the bacteria. And then they did a centrifuge, or they did separated out all of the different substances in the solution by layers. And what they found was that the layer where the viruses were found was no longer radioactive, but the part of the sediment that contained the bacteria also contained the radioactive material. And what that told them was the DNA that had been inside of the viruses was now inside the bacterium because the virus had injected the DNA into the bacterial cell. Now, just to confirm that this was really true, they did a second experiment where they actually made the protein capsule or outer covering of the virus the radioactively labeled part. And again, they allowed the bacteriophages to infect the bacteria. They went through the whole blender procedure. They centrifuged. And what they showed was that the radioactive part stayed attached to the viruses. In other words, that protein capsule stayed with the viruses. It was not the infectious agent. Rather, the DNA from the previous experiment was shown to be the infectious agent. And again, this just confirmed that DNA is the portion of the cell that is the one which controls the activities of the cell. It's where we keep all the information for the manufacture of proteins and other activities in the cell. Now, really quickly, let's go through a quick review of what DNA is made of. You will remember that DNA is a type of nucleic acid, and that nucleic acids are one of the 4 types of organic molecules. You'll also remember that nucleic acids are made of long strings of individual nucleotides, and that nucleotides contain a total of 3 parts in them. They contain a sugar group, they contain a phosphate group, and then they contain one of several different nitrogenous bases. And the type of nitrogenous base present will determine what kind of nucleotide you have. Now, we can divide nucleotides into 2 broad categories. You can either have a purine or a pyrimidine nucleotide. Purine nucleotides are nucleotides that are made of two rings containing carbons and nitrogens, oxygens, etc. And they include things like adenine and guanine. By comparison, pyrimidines, the nitrogenous base of a pyrimidine, or the pyrimidine type of nitrogenous base, sorry, is made up of a single ring of carbons and nitrogens with some oxygen attached. And examples of this include thymine, cytosine, uracil. Okay, so now remember again that the new types of nucleotides found in DNA include adenine and guanine, both of which are purines, and they include thymine and cytosine, both of which are pyrimidines. Uracil is found only in, if you remember, RNA. Okay, there's another experiment that's briefly discussed in your book, and I'm not going to go really over the details of it, but Chargaff's experiment was the experiment using radioactively tagged markers on these different nucleotides to determine the relationship of how these nucleotides interact with each other in DNA. And to make a long story short, what Chargaff found was that adenine always pairs only with thymine. Remember that DNA is a double-stranded helix, so one side of the double strand, if you had an adenine on the opposite side paired with it, you would always have a thymine. And the second thing Chargaff found was that guanine always and only pairs with cytosine. So we're still on talking about the structure of DNA. You'll recall that I just mentioned that DNA is a double-stranded helix, and there were two individuals who discovered this double-stranded helix by the names of Watson and Crick. They eventually received the Nobel Prize for that. I always make it a point to mention the fact that there was another individual whose research was absolutely indispensable to Watson and Crick being able to describe DNA as being a double helix. And that was the work of a woman by the name of Rosalind Franklin. She did lots of work on X-ray crystallography, and here's an X-ray crystallograph that she did of DNA. To make a long story short, she had been trying to figure out the structure of DNA as well, following a different line of inquiry from what Watson and Crick had been doing. She was using crystallized DNA molecules and then taking X-ray photographs of the structure of the DNA and trying to work out, was it a triple helix, a double helix, a single strand, what was it, by that approach. Watson and Crick happened to be visiting the university where Rosalind Franklin was working, and basically went and looked on her desk, saw this picture that Rosalind had taken of DNA, and intuited from that oh my gosh, we've been wrong all along. The structure of DNA is a double-stranded helix. When they published their paper, when Watson and Crick published their paper outlining the double-stranded nature of DNA, they did not give credit to Rosalind Franklin for having used her work to help them determine the final shape of DNA. Which is very unfortunate. She was short-shrifted. She later died of cancer, and as a result, by the time Watson and Crick won the Nobel Prize, Rosalind was not alive, and since they don't award the Nobel Prize to anybody after their death, she was never really truly recognized for her contribution to the structure of DNA. Okay, so some more specifics about DNA structure. Now, when we talk about DNA structure, you're going to find out that we're going to refer to something called the 5' and 3' ends of DNA. So I want to make sure I talk about what that means. If we look over here, here is a simplified drawing of a double-stranded DNA molecule. And you will notice, as I told you before, that A always pairs with T, C always pairs with G. You can see that relationship quite clearly here. Remember, these dashed lines here are representative of hydrogen bonds. It is hydrogen bonds that hold together C's with G's and T's with A's across between the two strands. Remember, hydrogen bonds are not true bonds. They are just attractions between partial positive and negatively charged regions of molecules. Notice too, if you would, that outside of where the A's pairs with T's and the G's pairs with C's, there is this alternating sugar phosphate, sugar phosphate, sugar phosphate backbone present. Okay, remember, a nucleotide has three parts to it. It has a sugar group, a phosphate group, and a nitrogenous base. The nitrogenous base is always oriented in the middle of the DNA molecule, like the rungs of a ladder that you step on and grab onto with your hands. The sugar phosphate backbones are like the sides of the ladder, okay, the up-down part of the ladder. Now, what I want you to notice is that if we looked at the very end of a DNA molecule, one of the two strands would terminate with a phosphate group. The end which terminates with the phosphate group we call the 5' end. And if we look on the other side, on the other DNA molecule over here, you will see that this end ends in a sugar group rather than a phosphate group. We call the one that ends in a sugar group the 3' end. So 5' end ends in a phosphate, 3' end ends in a sugar. You'll notice down at the other end of the molecule down here that here we end with a sugar group, so this is the 3' end of this strand, and here we end with a phosphate group, so that's a 5' end of this strand. Hopefully you notice that these two strands are what we call anti-parallel, meaning that one strand goes from 5' to 3', and the other strand does the opposite. It goes from 3' to 5'. Okay, that's enough on basic DNA structure. The next lecture we're going to talk about how DNA makes copies of itself.